Well, today I want to talk about something that's called post-transcriptional regulation of gene expression. When we think about gene expression, we think about genes that are present in the DNA and the chromosomes. The gene gets activated by all these various transcription factors that we looked at. The messenger RNA is made, and then the messenger RNA is translated into protein. So the DNA encodes the messenger RNA, and then we get protein. That's kind of the goal of the whole system. It's ultimately get a protein, which gives rise to some specific function that the cell needs at that time. And if we talk about regulation of gene expression, we often talk about, or people often think about, regulation of transcription. That is, you need to get a particular gene made, particular protein made, so you turn the gene on. This is quite common. Probably one of the more common ways, and that's referred to as transcriptional regulation. But we also saw when we talked last time about, or a few times ago, about translation, we saw something else. We saw that you can turn on and off translation through the phosphorylation of the initiation factor EIF2. And if the EIF2 is phosphorylated, then it can no longer function in the initiation complex for translation, and so you don't get the protein. So this is another level of regulation. This is referred to as post-transcriptional regulation. So I want to talk about that a little. Well, I want to talk about that today, and we're going to actually talk about three different types of post-transcriptional regulation. We're going to talk about regulation of translation, and we already saw one example of that, and that was through the modification of the initiation factor. And this topic flows right. It makes sense to talk about it after translation. But I also want to talk about regulation of messenger RNA stability. And then finally, after the protein is made, there's other levels of regulation as well. And we'll talk about a few of those, but one that I want to focus on today is regulation of protein stability. So the first thing I want to do is talk about the regulation of translation. So let's take a look here. So regulation of translation we're going to look at first. So I mentioned when I first described to you the structure of the messenger RNA, there's the 5' cap, the polyadenylated tail, the AUG, which is the initiation codon. I mentioned back then was usually not at the very beginning of the message, but it's usually somewhere towards the middle of the message. And then the stop codon is not at the end of the message. It's usually, somewhere towards the end, but it's not at the end. And so there's two regions that are called untranslated regions. There's the 5' untranslated region, 5' UTR, and then there's the 3' untranslated region, 3' UTR. And these UTRs are extremely important in regulating different aspects of the process. So the 5' UTR is generally required or is involved in regulating translation. What I have written there is regulates initiation. And the 3' UTR also is involved in regulation, but it's regulating messenger RNA stability, and we'll look at that in a little while. So let's start, first of all, focusing with the 5' UTR. So the 5' UTR is involved in regulating translation, and it does so by a number of different mechanisms. And one is that it is often a site for recognition by microRNAs, and we'll talk about microRNAs in a little bit more detail in a moment. But basically, the microRNAs are small RNAs. They're 10 to 15, sometimes a little bit bigger, 20 or so bases, and they base pair due to specific homology or base pairing capability with regions of messenger RNAs. And if, for example, it binds to the 5' UTR, that will block the scanning to the AUG. It would basically block translation. Sometimes it's also involved in the destruction of the messenger RNA. So if it binds somewhere else, not in the 5' UTR, or binds somewhere else in the middle of the RNA or towards the end of the RNA, it signals a reaction that cuts the RNA and basically eliminates it. So let's just look a little bit at these microRNAs. So here's just a short list of some of the properties of microRNAs. Again, they're small RNA molecules that control gene expression, anywhere from 10 to 20 or so nucleotides. There's been around 2,000 that have been identified. These are 2,000 specific microRNA genes. And in fact, there's evidence that there may be as many as 5,000. These are very specific genes in the genome. They have their own promoters. They're translated in response to environmental signals at different cell types under different conditions. They have extremely important functions. They do not encode proteins. They're involved in regulating gene expression, basically regulating the usage of messenger RNAs. They're made as larger RNAs, which are processed by a complex called dicer down to the size of around, here's this 22 nucleotides. And then that 22 base pair microRNA is bound to a series of proteins, which is called a RISC complex. And I have a diagram in a minute that I'll show you. But the RISC complex bound to that RNA then base pairs with specific messenger RNA molecules. And depending on where it base pairs, it either leads to repressing translation, so a way of regulating translation, or degradation of the messenger RNA. You can regulate the production of a protein by eliminating the RNA. You eliminate the messenger RNA, you won't get protein. And then the other point is that deregulation of many different microRNAs are responsible for lots of diseases. I'm not going to go into that, but I'll just have you know that there's a number of cancers that have been identified that are specifically due to the overexpression or, in some cases, underexpression of specific microRNAs. Because, for example, if you have too low of an expression, or let's say a deletion in a microRNA gene, then another gene or another family of genes will be overexpressed due to the loss of the repression of its translation or the loss of its degradation. So these are turning out to be extremely important. So this is just kind of a little schematic of the pathway. So in the nucleus, which is shown in kind of orange, there's a specific genes, or there's 2 to 5,000 specific genes encoding different microRNAs with different nucleotide sequences. And these are made as longer pre-microRNAs, which are then complexed with proteins in the nucleus that trim it down to a smaller molecule, which is internally base paired with this hairpin loop, but that is exported out of the nucleus into the cytoplasm. Once it's in the cytoplasm, which is in kind of yellow here, then it's complexed with that dicer protein that I mentioned and another protein. And what that does, it trims it down to the 20 or so nucleotides and eliminates one of the 2 strands of the RNA. It degrades it. And then that single-stranded RNA gets, which is now the microRNA, gets complexed into this RISC complex and then binds to a whole series of other proteins, which are shown in the bottom left, and then specifically through specific base pairing will bind to very specific messenger RNAs. That's kind of illustrated here. And then depending on where it binds, it has different effects. It can block, as it's showing here, it's blocking movement of the ribosome down the messenger RNA, so it halts translation. Or it can lead to messenger RNA cleavage, or basically cutting up the message. So what we're seeing here is an example, in a sense, of how these microRNAs can regulate translation, This is post-transcriptional, a mechanism of post-transcriptional regulation. It can turn on translation or turn off translation. Now I'm going to give you some specific examples, but we're not going to look at the microRNAs. We're going to be looking at some other aspects of the 5' UTR. So this is the same drawing that I showed you before. Again, we're focusing on the 5' UTR. And so I say down below the site for recognition by microRNAs, but it's also a site, or they can be sites, for recognition by specific proteins. And these proteins often bind to complex secondary structures in the messenger RNA. So often the 5' UTRs can undergo internal base pairing, which can lead to alterations in its activity, but it can also lead, can be binding sites for proteins that can bind to those stem loop structures. So I'm going to give you some examples of that. So we're talking about examples of regulation or regulation of translation through the 5' UTR. And I'm going to give you a couple examples, but the first example I want to give you is regulation of translation of messenger RNAs encoding ribosomal proteins. Now there are 88 different ribosomal proteins, and they're all encoded by specific genes and specific messenger RNAs. So there's a messenger RNA for each ribosomal protein. And these ribosomal proteins use the ribosomal RNA as structural support. So the ribosomes, as we've seen, are complexes of ribosomal RNA and protein. And the ribosomal RNA acts as a substrate for the binding of these different ribosomal proteins to build the ribosome. But as it turns out, their translation is regulated. So the translation can be turned off and can be turned on through the 5' UTR. So I just want to show you this one picture here. So this I'm just showing you to recall that these ribosomal RNAs fold into very complex structures through internal base pairing. So there's a prokaryotic ribosomal RNA, and then there's the eukaryotic ribosomal RNA. But the bottom line is, you can see, these undergo very specific internal base pairing, and the internal base pairing causes these very complex stem loop structures, which are extremely important. In one case, for the peptidyltransferase reaction, the structure of the ribosomal RNA leads to the activity of that enzymatic activity, which links amino acids together. It's extremely important. The RNA is enzymatic, but these stem loop structures also serve as binding sites for specific proteins. And then the ribosome gets built up in that way. So it acts kind of like as a skeleton. Now I'm going to give you one example, but there's many. I'm going to give you an example of a protein, the regulation of translation of a protein called ribosomal protein S7. So let's look at that. So here we're going to look at the specific regulation of translation of a protein called S7. It's a ribosomal protein. I'll describe in a second. But the regulation of the translation is due to the binding of a specific repressor molecule, repressor protein, to the 5' UTR of the messenger RNA. So let me explain a little bit of what's going on here. So the ribosome has around 80 proteins, 88 proteins, and they're called things like S1, S2, S3. That refers to the small subunit. L1, L2, L3, it goes on, L4, L5, L6. These refer to proteins in the large subunit. So we're going to be looking at, the example we're going to be looking at is the ribosomal protein S7. So it's number 7 of the small subunit. Now, these proteins, S1, S2, S3, S7, all bind to specific stem loop structures or specific sequences on the ribosomal RNAs. So that's what I'm trying to illustrate here on the left. So proteins bind to the ribosomal RNA to build the ribosome. So here I'm showing I'm showing the ribosomal RNA as a squiggly line. And then these different proteins bind to very specific sites on the ribosomal RNA, acts as a backbone, kind of the internal structure of the thing. Now we're talking about ribosomal protein S7, so people have studied that, and so the question is, where does it bind to on the 16S ribosomal RNA? And we know exactly where it binds. It binds between nucleotide 940 and 1340 on the ribosomal RNA. It's a very specific site, and if you look at that site, it forms a kind of a complex dual stem loop structure, which you can see there. So that the protein is recognizing that sequence, and that's how it binds. Well, it turns out that the S7 protein also binds to its own 5' UTR. So this is end product repression. The messenger RNA, which is shown in the middle, the S7 messenger RNA encodes the S7 protein, and that S7 protein can bind onto the 5' UTR and block translation. So it can regulate its own translation. So the assumption is, and what I've shown below, is let's look at the right-hand side. It says enough S7 in the cell. So the idea here is that if there is not sufficient ribosomal RNA in the cell, then the cell chooses, in a sense, doesn't really choose, but the cell then does not make more ribosomal proteins. It doesn't make sense to make the ribosomal proteins if there's no ribosomal RNA to bind to. So the observation is if there's enough S7 in the cell, meaning there's no free ribosomal RNA to bind to, then the S7, rather than binding to the ribosomal RNA, will bind to its own 5' UTR and block translation, turns itself off. If there's then synthesis of additional ribosomal RNA molecules, and that's on the left-hand side, so that means if there's ribosomal RNAs in the cell, then there's reason to make more ribosomes, and so then it turns on the synthesis of these proteins, and so that particular phone call, 1 second. Okay, sorry about that, I had to take that phone call. So back to where we were. So if there's enough S7 in the cell, meaning that there's no available ribosomal RNA to bind to, it turns itself off by binding to the 5' UTR. If we need more S7, then meaning that there is available ribosomal RNA in the cell, time to make more ribosomes, then that protein comes off the 5' UTR and then allows translation to proceed. So here you have an example of regulation of translation by binding to specific sequences in the 5' UTR to regulate the availability or the abundance of this particular protein. So people have asked, what is it about this 5' UTR that allows the S7 to bind? And so people sequence the 5' UTR, sequence the ribosomal RNA, because if the S7 is binding to the ribosomal RNA, then maybe there's a very specific sequence. And so looked at the sequence of the 5' UTR, there was no similarities whatsoever. And then people plugged in the sequence into a program that would generate secondary structures. And it turned out that the 5' UTR made a series of hairpin structures and stem loop structures that were very similar to what's seen in the ribosomal RNA itself. So here's what's shown here. So this is the model of the 5' UTR. So if you look at the top is the message. This is the message of the S7 messenger RNA. and you see the AUG, and then right upstream of the AUG is the stem loop, series of stem loop structures, and if you look at the binding site on the ribosomal RNA, it's almost identical. So it's not the sequence that it's binding to per se, but it's the structure that the S7 is binding to. And so the structure on the 5' UTR basically mimics the ribosomal RNA binding site. It's a little bit different, a little less affinity. So if there is ribosomal RNA available, the S7 has a higher likelihood of binding to it. But if there is no S7, if there is no ribosomal RNA, then it will bind to the 5' UTR. So it acts as a mimic. So it's a very interesting mechanism and strategy for regulating its own translation. Let me give you another example. Here's example number 2, and this is, it's kind of a similar mechanism in a sense, but the strategy is a little bit different. So here we're looking at the regulation of synthesis of an iron storage protein called ferritin. Ferritin binds to iron and it stores it. It keeps it available for when the cell needs it. So the ferritin is a protein, and you see up at the kind of the top right, it's the cap structure, the poly A, the UTR, and then there's the open reading frame, and that encodes ferritin protein. The ferritin protein binds iron. So the observation is, if there is high levels of iron, then there's an increase in the amount of ferritin protein. So high iron, the ferritin protein goes up. People looked at transcription of the gene, and the transcription of the gene stays the same. The abundance of the messenger RNA stays the same. What changes is the translation efficiency, so you get more protein by turning on translation. So there's increase in translation. Now at the 5' UTR, there's what's referred to as an iron response element, an IRE. Iron response element, and that region of the 5' UTR does form a stem loop structure, and it's absolutely required for this regulation by the abundance of iron. So when there's high iron, there's an increase in translation to make more ferritin to bind the iron, and if there's low iron, then we don't need to have ferritin in the cell, and so there's a decrease in translation. So how does that work? Works like this. I'm showing you below the messenger RNA. So there's the cap, the poly A, the AUG. There's a stem loop structure. And in this particular case, where there's nothing bound to it, it's active. The translation will go. But when there is low iron, that region of the 5' UTR is bound to a protein, which is a translational repressor, which is called aconitase, which I have circled in red. So aconitase is a repressor molecule that binds to the 5' UTR of ferritin and blocks its translation. You can see how it would do that. It would block scanning and the initiation at the AUG, so it's off. Now on the right-hand side, I show you what happens. So aconitase also has a binding site for iron. And when it binds iron, iron acts as an allosteric regulator. When it binds iron, it changes just so slightly the structure of this repressor molecule aconitase, and it allows it to come off of the 5' UTR, come off of that stem loop. It dissociates and allows translation on. So if you get an increase in iron, we need more ferritin. So there's an increase in iron, it binds to the aconitase, changes the structure, it comes off of the messenger RNA, and translation is turned on. So this particular slide just kind of gives an overview. Both of those examples, the S7 and also the aconitase repression of the Ferritin translation, is just like this. So you have the AUG and the 5' UTR. You have a stem loop structure, and the stem loop structure causes the binding of a particular repressor. And so when the repressor is bound, either it would be S7 protein or it would be aconitase, no translation, no protein made, and when that repressor comes off, then the protein is made. So these are examples of proteins binding to the 5' UTR regulating translation. I want to give you one more example, but this is not an example of protein binding, and it's actually a really interesting and fascinating example. So this last example I want to give you is regulation of translation of sigma 32. Okay, now sigma factors, these were the factors that bind to the E. coli RNA polymerase to give it the specificity. Sigma 32 is a specific sigma factor that is induced in response to heat. Then it binds to the RNA polymerase and then turns on genes that allow the E. coli to survive under that high heat. So what it says is required for heat stress response. Now, when bacteria are normally at 37 degrees, but when the temperature is elevated at 42 degrees, the sigma 32 appears. So the question is, how does that happen? Is it transcription? How would the specific transcription of sigma 32 appear if there's no specific transcription factors that are responsive to heat? And those don't get turned on until sigma 32 is bound to the RNA polymerase. So it turns out that the sigma 32 messenger RNA is present all the time in the cell. So it's made all the time, but it's just not translated. And it's only translated under high heat. So if you increase to 42 degrees, you get increased translation. And that regulation takes place through the 5' again, through the 5' UTR. So that's a temperature regulated translation. So how does that work? I'll show you an example. I'll show you a picture in a second, but what happens is that under normal conditions, the 5' UTR forms a complex stem loop structure, which blocks translation. And under high heat, it comes apart. So this is basically what happens. So this is the sigma 32 messenger RNA. It's referred to as a thermosensor. Sometimes it's a RNA thermometer because it's sensing temperature. So here you see under normal conditions at 37 degrees, you see the stem loop structure blocks translation. And at 42 degrees, it denatures, it opens up. So what that means is that the nucleotide sequence that makes up that stem structure has the right combination of A's and U's and G's and C's, because A's and U's will denature at a specific temperature and G and C will denature to specific temperature, higher temperatures. And so it has the right combination of those sequences to allow it to denature at 42 degrees, not 40 degrees or not 38 degrees, but at 42 degrees. And in fact, people have been able to engineer these things to denature at different temperatures, because if you change the GC-AU ratio, you can alter the temperature at which it will denature and then expose the ribosome binding site and allow translation to continue. So these were all examples of regulating translation, post-transcriptional regulation, regulating translation through the 5' UTR. What I want to talk about next is regulating translation, regulating gene expression through regulating the stability of a messenger RNA. Well, I'll explain that in a second. So here we have more on regulation of translation. Actually, we're talking about regulating the stability of the messenger RNA, and that occurs through the 3' UTR. So by stability, what I mean is how long the messenger RNA stays in the cell. So if the messenger RNA stays in the cell for a long time, then you get a lot more protein. If the messenger RNA stays only for a very short time, then you get very little protein out of that RNA. So I have a kind of a small table here just giving you some examples. So we have actin has a half-life of around 60 hours. Half-life means that after 60 hours, half of what was originally there at the beginning of the experiment is left. So after 60 hours, half of it's gone. So actin has a half-life of 60 hours. Globin, and it makes sense because actin, we need a lot of actin in the cell for all the for all the cytoskeleton. A globin, which is, generates the hemoglobin, has a half-life of 10 hours. C-FOS, which is a gene that is involved in the cell response to growth factors, has a half-life of just 10 minutes because it's only required as the cells are stimulated to divide. And then we have peptide hormones, which have prolactin and oxytocin. Those messenger RNAs only stay in the cell for 2 to 3 minutes. What happens with these particular hormones is that you get the signal to make a hormone, and the whole idea of a hormonal signal is that it should happen very quickly and then disappear. So oxytocin, for example, when oxytocin is induced when a woman is in labor, the oxytocin is what induces the contraction. So the gene gets turned on, the messenger RNA is made, but the messenger RNA only stays for 2 to 3 minutes, so you only get a small amount of oxytocin produced. And that, you might say, is a good thing, because you don't want the contraction to last for 10 hours. And that's what would happen if the half-life was 10 hours or 20 hours. So it's 2 to 3 minutes, turn on, make what you need, and disappear. Now, the stability of these RNA sequences are regulated through the 3' UTR. And specifically, they're regulated through a particular sequence, which is the sequence is AUUUA. Those sequences are present at the 3' end of UTR, depending on the messenger RNAs. And those sequences, the AUUUA, is a recognition sequence for an enzyme that cuts the RNA and then allows it to be degraded and disappear. And the more AUUUA sequences there are, the faster the messenger RNA disappears. Also, there's microRNAs that do bind to the 3' UTR and allow it to be cut as well. And so the turning on of a microRNA gene, you'll get the binding of that microRNA to the 3' UTR and get it cut. Now, the messenger RNAs that I've talked about here are pretty stable in terms of their half-life in the cell. But these half-lives, depending on the gene that you're looking at, these half-lives can be altered. That means that it can be regulated. It can be a particular RNA can have a long half-life and then be induced to convert to a short half-life. So here I want to describe something to you. So if the goal of turning on a gene, the goal of gene expression, is to generate the protein. That's the end product. So you can get increase in protein by different mechanisms. You can get increase in protein by changing translation. Obviously, we saw that. But here I want to look at two different examples. On the left, we have increased transcription. So if you have a situation, so on the y-axis, we're looking at the amount of messenger RNA, or it could be the amount of protein, but it's the amount of messenger RNA. And then we have the transcription rate going from 0 to some high level by turning on the gene by various transcription factors. Obviously, when there's no transcription, you get no messenger RNA, but as the transcription rate increases, the amount of messenger RNA increases as well. And most people, most scientists, in fact, if you are looking at the increased amount of protein in a cell, or even if you're looking at the increased amount of messenger RNA in a cell in response to some stimuli, most people will assume that it's increased transcription. In fact, that's why the transcriptional regulation has been so well studied, because there's an assumption that it's at the level of transcription, but that's not always true. The other way to increase the amount of messenger RNA, one is by increasing transcription, but the other is by increasing stability. So on the right-hand side, what's shown, you're looking at the level of messenger RNA on the y-axis, and then you're looking at stability of the message on the x-axis. It can go to low stability, or like minutes, half-life of minutes, or it can change to half-life of hours if the transcription rate is constant. What will happen is, as the half-life increases, the amount of the level of RNA will increase as well. My analogy is just a visual. If you have a sink, turn on the water, and the water is flowing, and it is, kind of going down the drain. And you can increase the level. You can increase the level of the water in the sink by doing two things. You can increase the level of the water in the sink by turning on the water higher. Turning the knob, make more water come out. That will eventually allow the sink to fill up slowly. So that would be analogous to increasing transcription. The other thing you can do is you can slowly close the drain, leaving the water running at the same rate, but closing the drain will also allow the water to rise. So you can get the same result by either transcription or changing the degradation rate. So this is another example of post-transcriptional regulation. Changes in stability of the messenger RNA result in elevated messenger RNA. So I'm going to give you a couple examples of that. So here we have an example, regulation of histone messenger RNA. Actually, I'm only going to give you one example because I think you'll get the point. Regulation of histone messenger RNA stability. So we remember histones. Histones are bound up in chromatin and the nucleosomes and all that. And we need an enormous amount of histones when DNA synthesis is ongoing. So during S phase, when all the DNA is being replicated, we need to have lots of histones to bind and make new nucleosomes and make new chromatin. So during DNA synthesis, that's during S phase, when histones are required, the messenger RNA half-life is 60 to 90 minutes. The transcription rate is constant. So the messenger RNA half-life during S phase is 60 to 90 minutes. When DNA synthesis is complete, and that would be in the G2 phase of the cell cycle, all the histones are used. We don't need more histones because the DNA is complete, chromatin is complete. What happens is the messenger RNA half-life of the histone drops to 5 minutes. So here we have regulated changes in the stability of the messenger RNA. And how that happens is at the 3' UTR, there's a stem loop structure, and that stem loop structure is bound by a specific protein, which stabilizes the message. And the stem loop protein, actually, the name of the protein is stem loop binding protein. Not a very creative name, but it's stem loop binding protein. That protein is bound to the message during S phase, protects the RNA, and allows the messenger RNA to have a 60 to 90 minute half-life, so you get a lot of protein made. Now, at the end of synthesis, at the end of DNA synthesis in G2, that protein gets phosphorylated. There's a specific kinase that comes in, adds a phosphate, and then that stem loop binding protein gets modified. It comes off the stem loop, and it allows that messenger RNA to become unstable. And if you look at the stem loop structure, if you look at the sequence, what you see is there's a stem, but the loop structure is the A, U, 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 A sequence that I mentioned was the target for these nucleases that cut the RNA. So when it's bound, stable, more protein. When it's unbound, comes off, and then the RNA gets cleaved at that particular point. So that is basically just an example of regulation of messenger RNA stability. And so you can see the different levels of regulating the gene. Transcription, you can regulate translation, and you can regulate messenger RNA stability. All these are highly controlled. Now, the last thing I want to talk about in terms of post-transcriptional regulation is the regulation of protein stability. So it's a similar kind of idea as the RNA stability. Some proteins have constant stability, some can be altered. So if you look at P53, well, here's a table showing a few different proteins. We'll look at P53. P53 has a 5 to 10 minute half-life. P53 is a tumor suppressor. It's involved in the response to DNA damage, and I'll talk about it a little bit more in a minute. Actin and tubulin, very long protein half-life, 7 to 10 days. Histone, which of course is required, for the chromatin, 250 days. It's getting close to a year. So very, very stable, but you can see different stabilities, different proteins. And these are inherent in the proteins. Now, changes in stability lead to changes in the level of the protein. It could be increased or decreased. Now, that's through a particular pathway, which is called the ubiquitin proteasome pathway. And I'll talk about that in a second. Ubiquitin proteasome pathway. Proteasome is a specific subcellular, it's not really an organelle, but a subcellular complex that degrades proteins specifically. So many proteins can change their stability depending on the situation. So let's give an example of P53, response to DNA damage. As I mentioned, P53 is required for how cells respond to DNA damage, turns on genes to induce apoptosis or DNA repair. Now, if you look at a normal cell, the half-life for the protein, P53, is 5 to 10 minutes. That's what I had in the chart. 5 to 10 minutes, very quick. So it's made, it's turned over, made, it's turned over. Now, if you hit the cells with UV light, which damages DNA, or any other compound that would damage DNA, X-rays, what happens is the protein level increases in the cell. So if you look at the graph below, we're looking at protein. So you have UV treatment on the x-axis that's treated for 6 hours, 12 hours, 24 hours. Or you're looking at the level of P53 on the y-axis, and you're following the P53 protein, and you're following the messenger RNA levels after treatment with UV. And what you can see is the dotted line represents the RNA. There's no changes in the level of messenger RNA for P53, but what you see is that the level of protein dramatically increases between 6 and 12 hours, and it continues after treatment with UV light. Now, that could be at the level of translation. You can have the situation where the RNA stays the same and the translation gets turned on. There may be some of that going on, but the majority of the increase is due to increased instability of the protein. Before I move on to tell you a little bit about proteasomes, because this happens through the proteasome pathway, as does most protein degradation. But what I want to point out is that let's just talk about the stability of the RNA with the A-U-U-U-A sequence, or the stem loop structures in the 5' UTRs or the 3' UTRs, or we're looking at the protein stability. All of this information, no, let me rephrase that. All these activities, all these properties are encoded in the DNA. They're encoded in the sequence of the messenger RNA. That means they're encoded in the DNA. So early on, when I talked about the different levels of information in the DNA, and of course we know about the protein coding sequences. And when you talk about information in the information content of DNA, most of the time people will be referring to the protein coding information, but you can see here there's lots more information encoded in the DNA. That the half-life of the messenger RNA is encoded in the DNA. The regulation of translation through the 5' UTR is encoded in the DNA. The stability of the proteins are encoded in the DNA. All this information is encoded in the DNA. That's why I said early on there's multiple levels of information present, and not all of it is easily understood. We understood the genetic code and the amino acids. That was an easy one, but there's lots more. Okay, so all this kind of degradation goes through the proteasome. So the proteasome is a large multi-protein complex that degrades proteins. And it kind of looks, under electron microscopy, like a kind of a hollow tube. And lining the hollow tube are proteases, which degrade protein, and there's thought to be around 15 different proteases that line the central cylinder of the proteasome. And you can see that on the right-hand side, where it shows the diagram with the yellow and the blue, the active sites of the proteases, central cylinder, and then there's at the top of the proteasome, there's what's called a polyubiquitin binding site. Now, ubiquitin is a 76 amino acid protein, tiny little protein. I mentioned it earlier when we talked about modification of histones by ubiquitination. So the way proteins are targeted to be destroyed is by the ubiquitination of the protein. But it's not just one ubiquitin, it's many get added, and that's why it's called polyubiquitin. There can be 3, 4, 5 ubiquitins added in a chain, and when those ubiquitins are added to the protein, it tells the cell this protein is targeted to be destroyed. We don't want it in the cell anymore. And so there's specific recognition sites on the one side of the proteasome which binds to the polyubiquitin, and then the protein is sent through the core or the central cylinder of the proteasome and basically gets completely degraded. As it says, the ubiquitin is not degraded, it's recycled, but as the protein moves through, it gets totally degraded into tiny little fragments, 8 or 9 amino acids. So that is how proteins get degraded, and that degradation can be regulated, and I'm going to give you an example of what happens with p53, because we saw that p53 in response to DNA damage increases the level in the cell due to increased stability of the protein. So here's the example, regulation of p53 protein stability. Now in a normal cell, a normal undamaged cell, what happens is that p53 is constantly being transcribed, RNA transcribed and RNA translated, constant level. But it's bound to a protein called MDM2. It's a complex of two proteins, p53 and MDM2. MDM2 is a ubiquitin ligase. So it adds ubiquitin, polyubiquitin to p53. And that targets it for degradation through the proteasome. That's normally what happens. Now, if the DNA is damaged, then that damaged DNA activates a couple different protein kinases. One is DNA PK, DNA-dependent protein kinase, activated in response to fragments of DNA, and then another protein kinase called ATM. So these two kinases are activated in response to DNA damage. Now, the protein kinases phosphorylate different proteins. The DNA PK specifically phosphorylates p53, and it activates it. But it's still bound to MDM2, but the ATM phosphorylates MDM2 and makes it inactive. So all these are allosteric conformational changes. So in response to DNA damage, there's dual phosphorylation. p53 gets, well, let's start with the MDM2. MDM2 gets phosphorylated. It comes off of p53, so it can no longer add ubiquitin ligase, or it can no longer add ubiquitin to the protein. And so the protein immediately gets more stable, and then the phosphorylation of p53 activates it to then bind to DNA. So this is a strategy for a very rapid response. Minutes. So let me just give you an example. If you had DNA damage, so DNA damage during S phase, let's say the DNA damage occurred towards the end of S phase. But you want to stop the cell from dividing. You want to either DNA repair or apoptosis, but it's at the end of S phase. It's like maybe 10 minutes or 15 minutes before the end of S phase. So if the regulation was at the level of transcription, then what would have to happen? You'd have to transcribe the gene. You'd have to cap and polydenylate it. You'd have to splice it. You'd have to get it out of the nucleus. You'd have to get it to the ribosome. You'd have to make the protein. And then the protein could be made active. So that could take hours. So here what happens is the protein's constantly being made. It's at the end of the process. And we want to quickly activate it within a few minutes. So what you do is it's constantly being degraded, and you inactivate the ubiquitin ligase. The protein that's being made is no longer ubiquitinated, and so now it increases very quickly. Literally within minutes, you get high levels of p53. So this is another level of regulation. It's post-transcriptional, but it's also referred to as post-translational regulation. Post-translational because it's after transcription, but it's also after translation. So it's post-translational regulation. Now, this last slide just shows all the different levels of regulation of gene expression, or where gene expression can be controlled. And you can take a look at this, and it's pretty much what I've just shown you, but I'll just walk through it quickly. So we have the DNA transcriptional control, turning off and on the transcription. Then you get the RNA transcript. You can have processing control. That means you can have different, if we're talking about the end product as protein, which I have on the right-hand side with the red arrow, that's the end product. Then you can alter the level of that product by RNA processing. That may be alternative splicing. Then another level of regulation and which we haven't talked about, which exists, is the messenger RNA transport, getting it to where it needs to get to, which is the ribosome. And it's not there right away. It takes time, and it can be sequestered in a particular location, or it can be brought to the ribosome. So that can be regulated. So then you have the messenger RNA, and of course you can have control of degradation, you can have translational control, then you get the protein made, and of course you can get many different levels of regulation. Even at that point, you can get degradation of the protein, so you don't have it. You get degradation of the protein, so it's not active, or you can get protein the activity control, which again could be through phosphorylation. We've seen that with the IF2. We've seen it with P53. That you can get inactive protein, you can get active protein, and that's referred to as post-translational control because it's after translation of the protein. Okay, so there we have it. Multiple levels of post-transcriptional control. That's what we talked about today. Okay, have a good rest of your day.